Today we're going through Khan Academy's MCAT Prep Cells section Prokaryotes Questions. All right, question number 1. Methicillin-resistant Staphylococcus aureus, or MRSA, is an antibiotic-resistant superbug that can cause deadly infections in humans. What would these gram-positive bacteria look like under a microscope? Okay, so gram staining. Clear rods, pink rods, purple spheres, or purple spirals. So, Staphylococcus aureus, so the coccus part means that it's circles, right?
Yep, exactly.
And then staphylo means they stack on top of each other?
Yeah, they cluster in staph.
Right. Whereas bacilli means that they like organize in lines.
Bacilli mean that they're rods, and
Okay.
strepto mean that they're in a line.
Ah, that's it. Strepto is in a line. Staphylo means they're like clustering on top of each other.
Yep.
Coccus, so they'd be clustering on top of each other, and they would be circles because it's Staphylococcus aureus.
Right.
So they're also gram-positive.
Yep, they're asking about gram staining here. Do you remember that process?
I don't, which is why I was trying to narrow down the answers by remembering stuff about the bacteria.
So you could and you would. So which one did you end up picking?
I would have picked purple spheres.
And you would be right, because in the gram staining process, which is actually kind of annoying sometimes, there's like 4 different reagents that you put on it. You heat fix your cells onto the slide, and then you do some sort of wash, and then you do your stain, and then you like clear it off with ethanol, and then you do iodine or something like that. And you would find at the end of that whole long process that gram negative ends up being pink, and
Okay.
gram positive end up retaining the purple because they have a thick peptidoglycan layer that holds on to that ink.
Okay.
So even when you wash your ink off, they still stay purple, whereas the gram negative have a much thinner peptidoglycan layer, and it's under a cell membrane, so the dye washes off of them.
I just remember all of my bacteria in the gram staining looking burnt.
Yeah, well, they are, so that's great.
Like very burnt.
Yes, yes.
I think I burnt all my bacteria every time I did it.
So I had one that was like hard for me to classify until I did an endospore staining one time, and mine actually looked black under a microscope, and we think I just used the wrong reagents. Yeah. But.
Okay, so purple spheres. So you could have really gone from either direction. If you knew that gram positive means it's going to hold on to more of the dye, it's going to stay purple, and then Staphylococcus aureus means coccus sphere. So purple spheres.
Yep.
And that is the correct answer. Okay, question number 2. Methanopyrus candeleri is an organism which lives in a hydrogen carbon dioxide environment. Hydrogen carbon dioxide environment. That's a weird wording.
Yeah.
And was first discovered in a hydrothermal vent where temperatures reach 230 degrees Fahrenheit. What sort of organism is this? Oh, okay. Well, that tells us it right there. So I work with these kind of things a lot because I do a lot of evolutionary work involving proteins that date back to Luca. So when you have an organism living in, when they say hydrogen carbon dioxide environment, I don't really know if that can give you much of a hint. However, hydrothermal vent, temperatures of 230 degrees Fahrenheit, that's an archaea.
Yep.
Thermophiles.
Yes.
I don't think any bacteria can make it that high, but there are some bacteria that are thermophiles, but for them that means like they get close to 100 degrees Fahrenheit, I think.
Yeah. So the only debate that Tal and I cannot get over is how to pronounce archaea, because all of my biology teachers ever have said archaea, and he said archaea, and I don't know. I just don't know if we can work with this, people.
I'm on the archaea train. Okay, question number 3. Which of the following is not found in both prokaryotes and eukaryotes? Ribosomes, cytoskeleton, double-stranded DNA, nucleolus. Nucleolus.
Yeah.
Gotta be nucleolus. And it's correct, yes. So prokaryotes, no nucleus, also no nucleolus.
Really quickly, explain to everyone what the nucleolus is.
I hope I remember this correctly because we just talked about this not too long ago.
Yep.
So the nucleolus is a dense region of the nucleus where the ribosomal RNA DNA is stored and then ribosomal RNA is synthesized.
Beautiful. Just
Awesome.
beautiful.
Perfect. You're going to get questions about that, by the way, people. Okie dokie. Question number 4. Which of the following statements about the systemic effect of bacterial cells are true? Gram-negative bacteria are more likely to cause systemic effects because their outer membrane protects them from several antibiotics, making treatment more difficult. Gram-positive bacteria are more likely to cause system effects because the peptidoglycan of their cell walls is tough and hard for medicine to penetrate. Gram-positive bacteria are more likely to cause systemic effects because the lipopolysaccharide in their membranes can act as a toxin to circulatory system, or gram-negative bacteria are more likely to cause systemic effects because their lack of an outer membrane means they can more quickly infect host cells. Systemic effects. So systemic means you're getting into the bloodstream and you're going all over the body.
Yep.
The infection is no longer local. So gram-negative more likely because their outer membrane protects them from antibiotics. Maybe. Gram-positive because the peptidoglycan in their cell walls makes medicine hard to penetrate. That sounds a bit more likely. But that doesn't explain why it would cause system effects. Gram-positive bacteria because lipopolysaccharide in their membranes can act as a toxin to the circulatory system. Okay, well that immediately says blood and it says toxin. Gram-negative because their lack of outer membranes. That's not correct. Gram-negative bacteria have an outer membrane. So, I'm gonna say, well, hold on. So, gram-positive bacteria, lipopolysaccharide in their membrane, they have a giant peptidoglycan cell wall. So, if there was a lipopolysaccharide in their membrane, I would guess that it probably wouldn't exert effects because it wouldn't reach all the way out. I might be totally wrong with that thinking. I'm going to say that... Oh, God, I have no idea. What are you thinking?
Okay, I'm just remaining quiet and grinning because I had micro way more recently than Tall did.
Okay.
So it's not even fair because I don't know how many years it's been, 3 or 4 or something since he's had micro. So LPS, one of my favorite things to talk about, is on the outside, the outer membrane of a gram-negative, and it can act as a toxin.
Mm-hmm.
And so that's something that's pretty dangerous about gram-negative. And so gram-positive, thick cell wall, hard for medicine to penetrate. Well, not necessarily. There's a lot of antibiotics that target the ability of bacteria to create cell wall. So it's actually really easier for us to treat gram-positive. And gram-negative, so the one that I ended up choosing was gram-negative are more likely to cause systemic effects because their outer membrane protects them from several antibiotics, making the treatment more difficult. Because that outer membrane protects the peptidoglycan from destruction from certain antibiotics like penicillin, I think.
Yes.
That cleaves peptidoglycan.
Yes. Oh my gosh, I remember that now. Penicillin cleaves peptidoglycan.
So there's not even a gram-negative one that talks about LPS, but I'm not sure like if there was one that said gram-negative and LPS, how we would have differentiated between A and C.
So this was a pretty good, for me at least, I would have had to have gone through this as a process of elimination, but that would be only if I knew that it's only gram-negatives that have lipopolysaccharide in their membranes, not gram-positive, and peptidoglycan is actually very easy for medicines to penetrate. That's what antibiotics tend to do, at least penicillin. So, yes, the correct answer was gram-negative bacteria are more likely to cause systemic effects because their outer membrane protects them from several antibiotics, making treatment more difficult. Because antibiotics want to attack the peptidoglycan.
Yes, exactly. And so their explanation for this actually addresses the question that I had. And it says that both of those things are true. So the outer membrane protects them, makes them more resistant to antibiotics and the host immune system because it actually, this is really interesting. Sometimes, actually, I think that's more viruses, so I'll mention that in a minute. But additionally, the LPS found on the outer membrane is an endotoxin that damages the host.
Okay.
Yeah. So actually, and I'll talk about the other thing when we get to viruses, so I'll just leave it alone.
Awesome. Next question. Question number 5. Which of the following is not a method bacteria use for genetic recombination? Conjugation, binary fission, transduction, transformation. The amount of times I've seen this exact question, I don't know. So let's run through these leading up to the ultimate correct answer.
Yeah, can you define them for me?
Oh, okay. So conjugation is when you have a sex pilus
Yep.
that transports a plasmid from one bacterium to another.
What plasmid would the I don't know how to classify the one that makes the pilus, but
Fertility factor.
Yes. And so what do
Yes.
we call that plasmid? What's the
F plasmid?
The F plus, right?
F plus plasmid.
Yeah.
Okay. So it's the one that is going to create the pilus has to have the F plus plasmid, which is the fertility factor.
Yes, exactly.
Okay, so transformation is when you have competent bacterial cells that pick up like random bits of DNA from their environment.
Or we can introduce it to them. It's a really, really useful wet lab technique. You dry lab person.
I know nothing.
And he used the term competent cells. Oh, I'm so proud.
Okay, and then transduction. Transduction is the natural transmission of genetic material from one bacterium to another via a bacteriophage.
Yeah, that like accidentally grabs some of the host and gives it to a different one.
Like when it was making its capsid, it accidentally snagged some of the host DNA or something.
That's what I'm assuming, yeah.
Versus transfection
Yep.
would be if we scientists designed some DNA that we wanted the virus to carry into a bacterium.
Yeah, and we use it all the time.
Right. So binary fission, that's it. That's the one that they want you to choose.
Yep.
Because binary fission, although it is more inaccurate than the way that we do it with mitosis, it is still not a significant source of genetic recombination. All right, and it's not an intentional source. Well, some might say that. Next question. Scientists have developed a dye for galactose, which turns darker shades of blue as the concentration of this monosaccharide increases. It's dose dependent. In an experiment using bacteria that can metabolize both lactose and glucose, researchers set up test tubes as shown in table 1, which is below. A plus sign indicates a sugar was added to the test tube. A minus sign indicates a sugar is absent. Assuming bacterial concentrations stay constant, which test tube will show the strongest color blue? Okay, so we have bacteria that can metabolize both lactose and glucose.
Mm-hmm.
If there's a plus sign, that means a sugar was added to the test tube. If there's a minus sign, it means it was absent. So test tube A, we have lactose and glucose. B, we have only lactose. C, we have only glucose. D, we don't have anything. And the dye for galactose turns darker shades of blue as the concentration of galactose increases. Well, which will have the strongest blue color? So lactose, if I'm not mistaken, is a combination of glucose and galactose. So I don't know if you could say between test tube A, which has lactose and glucose, and test tube B, which has only lactose, wouldn't they have equal parts galactose? Because galactose is just half of lactose. So I'm going to look and see, did they say anything about whether or not the fact that you're including lactose and glucose means that you have less lactose? I would say as the concentration of monosaccharide increases, dose dependent, I'm going to say that since the dye only changes color due to galactose concentration,
Yep.
that it's going to be choice B, where lactose was the only thing added.
Yep.
And that's correct. All right, let's look at the hints because, once again, people, we can't stress enough that if you're going through experimental design, like through passages or through even discrete questions like this, it's extremely important. And the MCAT wants you to be able to read experimental design, find the flaws, and understand exactly what the methods were. So, the lac operon that controls lactose digestion in prokaryotes normally operates at a very low basal level and is upregulated by the presence of its substrate. Lactose is a dimer of galactose and glucose. The absence of glucose leads to an increased cyclic AMP because now the cell must rely much more heavily on the lac operon for energy. Oh.
Yep.
So if you don't have glucose, you're going to be using more lactose, which means you're going to be making more galactose.
Yeah, I got this wrong the first 2 times I attempted to answer it. And because I was trying to listen to you and read it at the same time, I read the first sentence as scientists have developed a dye for glucose, which turns darker shades of blue. And so then I was so super confused. But then Tall does this thing that when he reads it out for you guys, that whatever passage that's given, he'll summarize it. So when I heard him start summarizing, he kept using the word galactose. And I was like, where did it even mention galactose other than like breaking down lactose? So reading is important. And so one of the things I think it wanted you to remember here was that glucose is preferential for a lot of organisms.
Yes.
But in those that can break down both, that have the lac operon, even though glucose is preferred, when it's not present, they will essentially use all of their, turn all of their attention basically to lactose to use that as its source.
Because you can cut lactose in half, have your glucose,
right?
But you will be producing almost as a waste galactose, which will then turn on this the lac operon or whatever. It'll at least make darker shades of blue for the dye that they're inserting.
Yeah, that's a good point to bring up. So the lac operon, we know, allows the bacterium to break down lactose.
Right.
So and we know that galactose is an epimer of glucose. Yes. So I wonder if it can be used because I wonder if there's an isomerase basically that can change it to
glucose.
Yeah, I wonder.
I don't know.
Anyway, good thoughts, Tall. I almost called you Small. Wow, it's getting late.
Okay, next question. What types of cells do you obtain after an F+, remember that's fertility factor, after an F+ bacterium conjugates with an F- bacterium? So what type of cells do you obtain? So, is it 2 bacteria with identical plasmid and chromosomal DNA, but one is F+ and one is F-? One new F- bacterium, which has transferred the plasmid to the other cell, which is now F+. 2 F+ bacteria with identical plasmid, but not chromosomal DNA, or 2 F+ bacteria with identical plasmid and chromosomal DNA. So, I don't think it's identical chromosomal DNA.
Good.
I know that one right off the bat. So the last choice is right out. I believe that they replicate their plasmid before, and they use the duplicate to go in.
That is a beautiful explanation, and it is something really cool to look at how they do it. So if you want to like Google this, that would be a really cool visual to help you cement it in your mind. They use rolling strand replication. So as they replicate it, it's just sending that plasmid right on into the other cell.
Oh, wow.
It's so cool. Oh, and the sex pilus, they like pull the other cell in closer, and that allows them to do rolling strand replication with like a really high efficiency.
Huh.
Super cool.
Okay, so based on that, yeah, it makes intuitive sense that you wouldn't just give up your fertility factor.
Right.
I don't know why. I mean, I guess I'm thinking about bacteria like they would be people, and like I wouldn't want to give up my fertility factor.
Oh no.
But yes. So the plasmid is replicated and then shot through the pilus to go to the other bacterium, which now has the F+ plasmid. So you would have 2 F+ bacteria with identical plasmid but not identical chromosomal DNA.
And they didn't even bring up the HFR, the high frequency like version of this, so I'm assuming we don't even need to know it. And I think we always say that the MCAT is a mile wide but an inch deep, and
Yep.
I think that's really a good example of it.
All right.
Understand the basic mechanisms of how everything works, but don't worry about too much detail.
Absolutely. Could not agree with that more. All right. Next question. Question number 8. Where does energy production take place in bacteria that can perform aerobic metabolism? Mitochondria, nucleus, inclusion bodies, plasma membrane. Aerobic metabolism. Now, bacteria, I'm about to sound really stupid or averagely smart.
Okay, go for it.
Bacteria don't have mitochondria.
Oh no. Did I just almost say that?
Membrane-bound organelles.
I just totally almost said that. I said aerobic. I saw aerobic metabolism, and I was like, oh yeah, endosymbiont theory. They totally have mitochondria. Ouch.
We live in the eukaryotic world.
Wow.
So even though we are technically mostly bacterium, so it's not mitochondria, it's not nucleus. I don't know what inclusion bodies are.
So not ethnocentric. I was being domain-centric.
Domain-centric, yes.
Oh no. I'm so sorry.
Getting meta with Psych Soch again.
We know what nucleus is. We know
Right.
mitochondria. And both of those are, have two membranes, so like totally membrane-bound organelles.
It's double not prokaryote.
Small. And plasma membrane. I'm gonna go with negative. Maybe inclusion bodies. I don't know what they are.
I'm gonna say plasma membrane because I think once again I don't know what inclusion bodies mean, but I
Ooh.
know that like cellular respiration always tends to happen on a membrane.
Yeah, you're right. Okay, so inclusion bodies. For some reason, I think that but it says aerobic. Okay, this is what I'm thinking. What inclusion bodies are.
Okay.
It may or may not be the right. It's just a definition.
Go for it.
I think that for bacteria that can use things like sulfur or other weird things for their metabolism, they hold that in the inclusion bodies.
Okay.
I think it's like a little pocket, but I don't think it counts as an organelle.
All right. I'm gonna say plasma membrane. 1, 2, 3.
But
I got it
Okay.
right. Okay. So let's look at those hints to find out what inclusion bodies are. Okay. Bacteria, like eukaryotes, rely on a concentration gradient to drive ATP synthase molecule. Bacteria are prokaryotes and do not have membrane-bound organelles. Energy production in bacteria takes place across the plasma membrane, with the gradient between the extra and intracellular environment providing the appropriate concentration gradient. Do you remember, so for everybody, I work on voltage-gated ion channels. Do you remember asking how and why do bacteria have voltage-gated ion channels? This might be it. I don't know for sure, but if you're keeping a
Oh my
concentration
goodness.
gradient of ions, this would make a membrane potential that might be useful in generating ATP.
we were just talking about that.
I think that's it. I'm not entirely sure. Someone might correct me. Tweet us at the Twitter we don't have. Next question? Or, oh no, we're looking up inclusion bodies.
Yeah, I just want to know what they are.
What have we got? So we have purple bacteria may cause cellular, blah, blah, blah. Well, we'll get back to you guys if we can figure it out.
Yeah.
Question number 9. Porphyromonas gingivalis is a gram-negative bacterium found in the oral cavity. It is gingivitis.
Oh.
It's found in the oral cavity. It's negatively affected by the presence of oxygen. What term best describes this bacteria? So it's negatively affected by the presence of oxygen. Is it a facultative anaerobe, an aerotolerant anaerobe, an obligate aerobe, or an obligate anaerobe? So if it's negatively affected, so obligate aerobe means you love oxygen, you have to have oxygen. Aerotolerant means, meh, it's no big deal. Facultative means, oh, that's nice, I might use that.
If I needed to.
If I need it. I mean, I love this lactate stuff, but I might use it if I need it. And then there's obligate anaerobe, which means it is obligated to not have oxygen.
That was very well done.
Yeah, I felt good about that explanation, and we got it right.
Okay, so just hear me out for a second.
Okay.
Okay, so I got it wrong the first time I put this down.
Mm.
The microbe person over here got it wrong. I'm embarrassed. I said aerotolerant because I was like, it didn't tell me that they die if they're in the presence of oxygen. But I guess if they're aerotolerant, then maybe they're just not even negatively affected. They just like ignore it.
Yeah, it's just another thing there. It's kind of like if we were in a like if we were in fog or something.
Yeah.
Like it's a high concentration of water vapor.
Right.
It doesn't do anything to us.
Right.
It's just there.
Okay.
Yeah.
I can hang with that.
Okay. Last question. Which of the following is most responsible for bacterial cell motility? Is it pseudopodia, pili, cilia, or flagella? It's not pili.
Not. I don't know.
I don't think it's cilia. I think cilia, like their purpose is to like grab things and to create more more membrane surface area.
Yeah. So that diapedesis, that cell walking.
Yeah.
So is that caused by pseudopodia, and how does that differ with flagella? You know.
Yes. So so diapedesis, I know it in terms of like the immune system.
Yeah.
When you have
bacteria. Oh my gosh. I'm not
Right.
even reading English.
So I don't know if bacteria have pseudopodia,
Yeah.
but if they did, I don't think it would be terribly. So I know amoebas. I've seen like videos of amoebas that
Oh,
like chase
yeah.
bacteria around.
Yeah.
I've also seen that with neutrophils. They use pseudopodia. They just like jut out a piece of them, and then the rest of it follows, and then it like keeps following the bacteria until it just eats it.
Nice.
So flagella. I think it's got to be flagella, and we're right.
Well done.
And I got a confetti party! Oh, Khan Academy is so good at making me feel good about these just little things like a confetti party. Good on you, Sal. Good strong word, gang.